And it's my pleasure to introduce Liviu Mirica. I apologize if I said your name incorrectly, but he is the, I hope I get this right, the William H. and Janet G. Lacan Professor of Chemistry at the University of Illinois at Urbana-Champaign. So not so far from where I am at present, previously at University of California, Berkeley. His interest, first interest is in synthetic, inorganic, and organometallic chemistry applied to renewable energy catalysis and oxidative organic transformations. But today he's going to talk about something else, another interest, which is development of bifunctional therapeutic and diagnostic agents for amyloid peptide disorders like Alzheimer's disease, and the study of the role of transition metal ions in neurodegenerative diseases. His talk today is Theranostic agents for metal A beta peptide interactions in Alzheimer's disease. Take it away.
Thank you very much for the introduction, Stephen. Hopefully you can hear me okay.
Everything's good. Yeah.
I'm going to start sharing my screen. You should be able to see the right screen, the title slide, and my laser pointer. I'm going to look in the camera now. Thanks again, Rams, for organizing this great symposium and also for inviting me. I've actually learned a lot from both the experimentalists and the computational people that have presented throughout the symposium today. And now I have the chance to tell you a bit about our work. As you see, we are a synthetic lab and we do different types of syntheses, but in this context, we like to be able to, in the long run, develop molecules, and I'll show you many of them are either metal binding or metal containing molecules as potential therapeutic or imaging agents for neurological disorders. In this case, we'll talk about Alzheimer's disease. So even though it's a very general talk or general title, I'll try to focus on things which are definitely related to metal peptide interactions. It's great to be among the last people to talk. I don't have to spend too much time on the introduction and introduce Alzheimer's disease to you. So I'm going to jump right into a very complicated slide, which is taken from this reference right here. This is just to put a bit in context, Alzheimer's disease, and especially the great efforts in trying to develop new therapeutic agents for Alzheimer's. I think Bernard earlier today has done a great job of showing the big problem, the big need we have in the context of developing new therapeutic agents for Alzheimer's. The last molecule to be approved as an AD therapeutic agent was memantine in 2003, which was really a long time ago in the context of new pharmaceuticals. And now, of course, if I'm showing you here in the schematic, if we just look at this amyloid precursor protein, this kind of squiggly line shown here in the center, which exists in all cells, especially in neurons, when it's being digested, there are several secretases that create different fragments. One of these fragments is the amyloid beta, A beta peptide. And this is really the culprit of forming amyloid plaques, amyloid aggregates, large ones, small ones, oligomers, protofibrils. There are a million terms for these aggregates. But really, many of the potential therapeutic agents have been focusing on either inhibiting these secretases, these hydrolases. So base inhibitors, gamma secretases, many of those are small molecules. They're unfortunately very not successful. That's why we don't have any of these inhibitors on the market. And lately, in the past maybe decade or so, we had many examples of monoclonal antibodies targeting these smaller aggregates, these oligomer A beta aggregates, oligomeric aggregates, such as some of these names shown here. Aducanumab, the one I'm going to highlight now, is of course now called Aduhelm. They've changed the name, and it turns out that helped to get it approved. But really, this has happened back a few months ago when this Aduhelm was approved as a potential therapeutic strategy for Alzheimer's disease. But I'm sure if you heard on the news, there are many discussions and controversies about whether or not this is a really good agent as a drug for Alzheimer's. So of course, in the context of small molecules, if one could develop A beta aggregation inhibitors, they're not going to be large molecules like antibodies. They have a chance of crossing the blood-brain barrier much more easily. You don't have to have these biweekly infusions of antibodies. and in the end will be a much less expensive therapeutic strategy for Alzheimer's. Now, of course, it's very hard to develop a small molecule therapeutic for especially CNS disorders, and in particular Alzheimer's, which is so complex and usually is associated with many other age-related processes. So why would an inorganic chemist like myself be interested in this field? Well, it turns out early on, people have discovered, and again, we heard that earlier today, that metal ions such as copper, iron, and zinc have been shown to bind quite tightly to these amyloid plaques, and you can find appreciable concentrations of these metal ions in amyloid plaques. I'm sure here again, by now everybody knows almost by heart the amino acid sequence of A beta peptide. I never remember it, but it's shown right here. It's characterized by a very hydrophobic C terminus, and that's what leads to this aggregation of this very insoluble really peptide. But it turns out the N terminus has quite a few hydrophilic or charged amino acids, especially ones highlighted here could bind to metal ions because they're either carboxylate type residues or the histidine, they have imidazole or tyrosine, quite a few of these classical metal binding amino acid residues. And early on, going back to the great work by Ashley Bush, has been shown that metal ions such as zinc and copper can induce the aggregation of these peptides, especially in this case, A beta 40. So now what are we trying to do? Well, it turns out that there are many factors that play a role in the pathology of Alzheimer's disease. I have here a great image that's so complex because it shows all the complex pathologies of Alzheimer's disease. This is taken from a very exhaustive review by Liviu from a couple of years ago, showing the potential role of various metal ions in the context of several pathologies of amyloid beta or Alzheimer's disease. I'm not going to look at every single of these processes, but really what we're interested in, let me go back for a second here. If you're looking at this kind of interaction of the metal ion pool, whether copper or iron or zinc, interacting with A beta peptide, whether it's the monomeric form or the oligomeric form or the fibrillar form, these interactions could lead to many deleterious processes such as reactive oxygen species formation, oxidative stress, as well as maybe promoting the formation of various types of these aggregates, especially those neurotoxic aggregates. So that's really what we're interested in. In the long term, we like to develop molecules. We call them bifunctional compounds or maybe multifunctional compounds that could bind to metal ions as well as interact with these various A beta species in a way of trying to control this process and potentially use this as a therapeutic agent. So that's really what we're interested in the long term. Now, today, I'll spend a bit more time actually on sort of the first step in this process, and that is trying to understand at a detailed level the interactions of A beta species with transition metal ions. And then I'm only going to show you one example on somewhat of a successful compound that we have developed recently. And then hopefully using some of these compounds briefly, I'll describe how we like to use these compounds as potential metal-containing imaging agents for A beta aggregate. But really very early on, more than a decade ago, we were curious to see what happens when we take the A beta 42 peptide, the longer, more neurotoxic species, and how does it interact with metal ions such as copper and zinc. At least these are the two ions I'll show you today. So we use classical biophysical biochemical assays. We'll use the classical thioflavin T assay to see whether or not, for example, various concentrations of metal ions such as copper affect the aggregation process. And early on, we noticed right away that when you have presence of metal ions, in this case copper, and larger amounts, you see this decrease in the fluorescence intensity coming from the THT binding to its fibrillar structure. So you see that even substoichiometric amounts of copper could lead to this decrease. Now, of course, you may say, well, copper is paramagnetic. Maybe you just have a paramagnetic induced quenching of this fluorescence. So you use other biochemical assays, for example, use native gel and Western blotting to show that when you have no copper presence, this panel A corresponds to condition A on the bar graph, shows that when you don't have copper present, you tend to form these large aggregates. Whereas when you have copper, in this case, one equivalent of copper in panel B here, shows that we stabilize this smaller aggregate. This is confirmed by TEM, transmission electron microscopy, showing that the typical fibrils formed in the absence of copper, will actually show much smaller aggregates, almost no aggregates, or very tiny aggregates that cannot be shown by TEM. So we came up with the conclusion that maybe what happens in this case that copper may be stabilizing these smaller aggregates, these soluble oligomers. Now, would this have an effect on the toxicity of these species? And indeed, we've been able to show that, for example, if we prepare oligomeric A beta species, this bar graph, so here bar number 2, the oligomeric species of A beta 42 is quite neurotoxic, whereas the fibrillar form is actually not that toxic. Most importantly, when you have copper present, along with even monomeric form of A beta, since copper seems to stabilize these oligomers, we do have appreciable toxicity of these, what we think are copper-soluble oligomer aggregates. So what we think is going on is that copper actually increases the neurotoxicity by stabilizing the soluble A beta oligomer species. By comparison, zinc also seems to reduce neurotoxicity, but we think it reduces because it forms these non-fibrillar amorphous aggregates. So slightly different mechanism. Why am I mentioning this? Because we wanted to dive a bit more into how is this process, how is this happening? How come copper could slow down the fibrillization or increase the stability of these soluble oligomers? So this is another study we've done quite a while ago using some more direct approach of understanding how copper addresses this interaction. So here we use mass spectrometry, mass spec, but we use the approach of using hydrogen deuterium exchange, or HDX, to pretty much probe the extent of aggregation. And we use this pulse method, or where at different time points, we can take an aliquot of this aggregation solution, and we can pulse it at a low pH. We treat it with pepsin, which automatically quenches the aggregation, but also cleaves our A beta peptide to 3 different peptide peptides. They turn out to nicely map the hydrophilic, the center region, and the C-terminus region. And then we can analyze the extent of deuterium incorporation onto these peptides. That is, the more deuterium incorporation you have, the less protection you have. That is, you form less aggregated structures, and the less deuterium incorporation you have, the more protection you have, because you made these tertiary structures or quaternary structures even in this case. So let me show you briefly what we noticed quite surprisingly. What I'm showing you is this level of protection in the presence of just A beta 42, and this is done under these particular conditions. It turns out when you add copper in a 1-to-1 ratio, we're surprised to see this sort of stable level of protection. And it turns out this seems to map nicely to the formation of these oligomeric species. By comparison, we can also do this thing at 37 degrees with stirring, in this case, and we see the same effect happening, except that, of course, now everything aggregates much more rapidly. So why is this important? Because now this allows us to actually look at how could a compound control this process. So we have a better way of looking at how compounds could control these copper-mediated stabilization of oligomers. By control, just so with the control here, the fibrils have a much larger extent of protection. So indeed, we're looking here at oligomeric structures. And it turns out, to take some, to not spend too much time on it, it turns out that different regions of these peptides, especially these different peptides, seem to aggregate at slightly different time points. And we've been able to show that this is statistically significant. And without going into the details of these experiments, that seems to be changed when you have copper present or you don't have copper present. So what was the take-home message from this study is that we think we have to develop compounds that will ideally interact with these oligomeric species when interacting with metal ions such as copper. So in this context is what kind of compounds we're looking at. We use this bifunctional approach, and here I'm showing some of our design principles. These are classical molecules that either bind to A beta species or classical metal binding compounds going all the way to chloroquinol, developed by Ashley Bush and coworkers. But really, we like to bind this or combine these regions. Let's call them the red fragment and the blue fragment, meaning the A beta interacting fragment and the metal binding fragment. And of course, many more have contributed to developing such compounds throughout the years. So here, let me just show you a few of these compounds. These are the very first compounds we've made. We've made many more since then. We tend to make these bifunctional compounds. We always characterize how tight they bind metal ions. Some of these compounds can have very high affinity for copper. Peak copper level is higher than 11, let's say. They have quite high affinity for A beta fibrils, affinities in the low nanomolar range, and they do tend to control the aggregation of A beta species. It turns out the very first molecules we made, actually these two structures right here, L1 and L2, were great at breaking down the fibers. However, they're actually, they led to quite toxic soluble oligomeric species. So this may led us to develop a slightly different approach in which we like to make molecules that do not break down the fibers. If anything, they should promote the fibrillization. We really want to stay away from these oligomeric species, which are really the most neurotoxic species. So this actually complicates a bit how we design molecules because we don't have a molecular model of these oligomeric species. There's some models out there, but it's usually a combination of theory and experiment. So it's hard to know exactly what is the true structure. And of course, everybody knows that these oligomeric species are extremely heterogeneous. So I'm only going to show you one compound here to show you kind of how we think about this compound. So in a nutshell, what we have learned over the past decade or so, it turns out that if we use molecules that seem to be amphiphilic, that is, they have a region which is quite hydrophobic, but they also have a region that's quite hydrophilic, those seem to bind with quite high affinity for soluble A-beta oligomers. Now, it so happens, you see my color coding here, the hydrophobic region happens to be A-beta binding fragment. The blue fragment here, which is the hydrophilic one, happens to be sometimes metal binding structure. We may not need to use it for metal binding, but it turns out we'll have beneficial hydrogen bonding properties that we think confers his amphiphilic nature for this molecule. Here I have some of the design principles for this molecule. I'm not going to go into it. They either are classical molecules that bind to A-beta species, or they're metal binding fragments or imaging agents for amyloid species. I'm going to introduce a few slides later the precursor to this LS4 compound. You see it's only the red fragment, that is only the amyloid binding fragment without that microcycle attached to it. And you see that's an important fragment that is required for the proper functioning of this molecule. So this molecule is fluorescent. We can use its own fluorescence to determine how tight it may bind to this A-beta species. In this case, I'm showing you how it binds to either A-beta oligomers or preformed A-beta fibers. We're surprised to see an appreciable turn-on fluorescence for oligomers. We can actually determine directly the KD values for this compound. And we're surprised to see that we see appreciable affinity around 50 nanomolar for both oligomers as well as A-beta 42 fibers. So this was exciting. So now we're trying to understand what are the other properties. Of course, in this symposium, we talked about different interactions between metal ions, especially redox active metal ions, and these compounds. We've been able to show that this compound, LS4, even the precursor, has quite powerful antioxidant activity. In this case, we use this Strollock's equivalent antioxidant capacity. And it turns out that both these compounds, they do quench radicals, in this case, the ABTS radical in this protocol. And really, we think it's due to that 2-methoxyphenol fragment, which is quite electron-rich, and is present in both of those structures. It turns out we also looked at metal-mediated antioxidant properties. In this case, we performed this copper-induced ascorbate consumption assay. So here I'm showing the results for the LS4, where it's in the presence of just copper and ascorbate, or copper, A-beta, and ascorbate. And you can see if you don't have any compound present, ascorbate gets consumed very quickly by copper. When you add LS4, it quenches that consumption or the oxidation, or you can add LS4 after you've started this copper ascorbate oxidation, and you can definitely inhibit. The same thing happens, although to a slightly lesser extent, in the presence of A-beta 42. Impressively, when you use the precursor, which doesn't have that metal binding fragment, we don't see such as a quenching of the metal-induced ascorbate consumption, which I guess in the end, it makes sense because we don't have that metal binding fragment. So now we wanted to understand better what's going on in this case. So we collaborate with Emad Taikorshid, an expert in molecular dynamics, and we try to see what happens when we dock this compound to, for example, the NMR structure that was recently published by Karuya and coworkers. And this is an A-beta 42 tetramers. And we see the interaction of our compound not only through the hydrophobic region, but also through our hydrophilic azomicrocycle. We've done the same type of simulations by using our pre-LS4 compound without the TACN. And you definitely see that we're missing quite a few of those interactions coming from that TACN microcycle. And it turns out if you measure the affinity of this pre-LS4 compound, it has very low affinity, higher than 10 micromolar KD values. And also we can quantify these interactions and showing you that actually the van der Waals interaction are quite similar is the electrostatic interactions that come from this TACN microcycle that seems to be extremely important for binding to this A beta species. The most exciting part is that actually this compound, because it's still fairly hydrophobic, can cross the blood-brain barrier, can actually bind to the A beta species in vivo. So here what I'm showing you, if we treat 5XFAD mice with LS4, and then after 10 days or 30 days, we analyze the brain sections from this mouse, we see that actually LS4 has accumulated and accumulating the regions that have high amounts of these A beta species, whether random A beta aggregates of different types or specific oligomers that are found in vivo. So definitely we think this compound seems to bind in vivo to these A beta species and seems to actually promote the reduction of the overall amount of A beta species in vivo, but also seems to have some downstream effects in the context of activated microglia. I'm showing you some quantitative data showing you that you have less what we think neuroinflammation in presence of our compound. And the same thing is true in terms of p-tau aggregates surrounding these A beta species that seem to be reduced when we treat the mice with our compound. So really, we're really excited about this molecule. This idea of amphiphilic structures is quite important for potentially developing new therapeutic agents. And in the last just minute or so, I want to show you how we think about using some of these compounds we've made that are known to bind metal ions, but try to use them as an imaging agent, in particular, positron emission tomography imaging agent. It turns out there are quite a few imaging agents that have been developed and actually FDA approved, yet they seem to use carbon-11 F18 radionuclei, which actually have fairly short half-life. So our idea is actually to take a molecule, usually the molecules we make, maybe that bind very strongly copper, and then try to preload them with radioactive copper-64, and hopefully that's how we develop our diagnostic agents. Over the years, we developed a few classes of this compound. Some of them are actually quite promising in vivo. They show specific binding to a beta species, but it turns out we can even take our LS4 compound, pre-radiolabeled with copper-64, and it turned out without going through all the details of this chemistry, in the end, we can actually perform brain uptake biodistribution studies, and we'll be able to show that our preloaded copper-64 preloaded compound seems to accumulate to a larger extent in APP mice than in wild-type mice, as shown here in the statistically significant higher uptake in these ID mice. So really think that we can use maybe this compound also for potential imaging applications in the context of Alzheimer's, but hopefully also we can extend this to other neurological disorders that have these amyloid aggregating type proteins. So with that, hopefully I've been able to show you that we can develop molecules that do take advantage of these metal A beta interactions, and we can use those compounds as potential therapeutic agents, but also they could be used as potential imaging agents, longer-lived imaging agents, whether it's for AAD or other neurological disorders. And with that, I'd like to to thank many of the people that have helped us do this work, our collaborators, as well as people I have highlighted here throughout the years, graduate students, postdocs, and undergraduates, and the funding sources for the funding. And of course, I'll thank you for your attention. I'll be more than happy to take any questions you may have. Thank you.
Thank you for a very interesting talk, and thank you for being absolutely right on time. So we have a few questions. Let's see. First, from Nicholas Fuller. Are these radioactive imaging agents a concern for causing disease or making it worse?
It's a great question. So I kind of went very quickly over that in order to be on time. The big difference, and actually we try to always maximize our efforts, it turns out for imaging applications, you have to use much lower concentrations of these imaging agents. So let's say, just to pull in the ballpark, maybe you need a least micromolar or high micromolar concentration for therapeutic agents in vivo. For imaging applications, especially PET imaging applications, nanomolar concentration may be enough. So it turns out we use very toxic molecules. So some of these big molecules we make, they turn out to be extremely toxic just by themselves to the cells or even to mice. But when we use them for imaging applications, we reach much lower concentrations. So you could say that toxicity of the molecule itself is not gonna, and also the same thing is true about the copper itself, the radioactivity of the copper, since it's still gonna decay fairly quickly, is not gonna have a direct effect People use in vivo quite longer-lived radioisotopes for other applications, and they don't seem to be that toxic, of course, in the long run.
Okay, thank you. And from Jeff Brender, oh boy, they're popping up all over the place. Do you see any difference in the biodistribution of your 64 copper PET imaging agents versus the plaque imaging agents in use?
Excellent point. Quick answer, we're definitely not there yet. The FDA-approved imaging agents, they have quite high brain uptake. So those percent ID per gram for those FDA-approved compounds, they tend to be in the order of 4, 5, 6% ID per gram. We're barely breaking 1%. But of course, one could imagine doing some sort of medicinal chemistry type approach or SAR studies to improve those that uptake. We think we are within range, but we're definitely not there yet. And the same thing is true in terms of trying to see where they go. Some of the molecules, they do lose copper. They're not the best chelators. Some of them are better than others. So that's what we work in improving these sort of pharmacokinetic type properties.
Okay, thank you. And then a question from me was, do you have a hypothesis to account for the huge difference you see between copper and zinc? One is oligomer promoting, one is fibril promoting. And related to that, can your compounds discriminate between copper and zinc?
It's a great point. We need to get back to it. We need to do more basic coordination chemistry studies on this. So definitely that's one thing that's on our plate. I can tell you the very first molecules we made, those two molecules, they seem to have about 3 orders of magnitude higher affinity for copper over zinc. Was definitely not like the compound that Bernard showed with 10 or 11 orders of magnitude difference. That's why I was curious how or what he thinks is the reason for that. But that's a great point, because definitely there's more zinc present in vivo than copper.
Okay, question from Tim Storr. Is the molecular modeling predictive of how these compounds change the amyloid aggregation process? So I guess it's a related question.
It's a great question, Tim. That's what I was asking Birgit about other MD simulations out there that can either take a copper complex and bind, or do MD studies on binding the copper complex or any metal complex, really, to bind to these fibrillar structures or soluble structures. Or could one try to see if you mix in silico a beta species, a metal ion of choice, and a compound of choice, can you see what's going to happen? So I'm really interested in talking more with MD folks and see how can one think about it. I could imagine seeing quite interesting observations out of it, but really we're just starting doing more of these MD. We are really babies in that regard.
And I think we have time for one more quick question, which is, again, from Jeff Brender. Is the binding competitive with THT? Scaffold seems similar, which may suggest a similar binding site.
Yeah, great point. Depends on the structure, depends on that red structure. If you have these types of structure, we have these steel beam type structures, or two phenylbenzothiazole structures, they seem to have the same binding affinity, the same binding site to THT. How do we know that? We can do a KD direct binding affinity, or we can do a THT inhibition binding affinity. And in some of these cases, we get very similar Ki and KD values. Some other molecules, which I haven't shown here, they seem to not bind the same site, or we get pretty random data when we do these THT inhibition assays. So it depends on the A beta binding fragment.
Okay. Thank you very much. And I think we need to move on to the next talk.